From the north side of the San Francisco Bay to Singapore, welcome to Urban X Real Talk Fitness Radio with your host, business owner, lecturer, author, master trainer, Tiaja. With over 30 years of experience in the health and fitness industry, he will challenge the fitness between your ears. So prepare your mind, body, and soul for the revolution of self-care, the evolution of fit, with real talk about real people, real health, real fitness, and the real deal behind our present illness culture. Real talk every time, all the time. Get weekly insights on how to shift your thinking, emoting, eating, training, hydrating, goal setting, and resting for you, the everyday athlete. You can cheat your fitness, but you can't steal your health flow. It's Tuesday, 9 a.m. Let's flow.
Welcome to Urban X Real Talk Fitness Radio, where we challenge the fitness between your ears. I'm your host, Tiaja. Why do we age? Is it because our cells possess some internal clock so that when the sand runs out of the hourglass, per se, we die? And what does aging look like? Well, wrinkles in the face, brown spots on the hands. Does age appear with the graying of the hair or the dimming of one's sight? Or is aging something more complicated and intricate altogether? Are the so-called signs of aging nothing more than symptoms of something else going on deep inside the cell? You know, one of the most intriguing arguments for aging I've ever heard was advanced by a woman by the name of Judy Campisi. When Judy Campisi, a leader in the field of cell senescence, gave her first lecture on the topic at Gerontology conference in the late 1980s, she received a scathing response from her peers in the scientific community. What she advanced back in the 1980s is still considered radical by many today. Cancer and aging, she observed, seem to be two sides of the same coin. Aging, she said, is the process or the price we pay for protection against cancer, and that nature's way of limiting proliferation of potentially dangerous cells is to put a brake on their ability to divide after a certain length of time. Now that's mind-blowing. No wonder her lectures were constantly met with acrimonious debate, but think about what Miss Campisi was saying. In essence, she was saying that our bodies have within them a mechanism to trigger a form of cellular euthanasia, or suicide if you will, in an effort to suppress the proliferation of tumor cells. The hostility towards her theories only began to subside as newer evidence mounted that clearing out senescent cells in a whole range of model organisms extended the lifespan by up to 30% and alleviated so-called age-related diseases. But was Miss Campisi right? Was this nature's way of neutralizing cells that might with time have gathered dangerous mutations over the course of time? Well, consider the science. Senescent cells have proved to be extraordinarily stable. Rumor has it that one lab in Texas has kept senescent cells perfectly happy in a culture dish for years until the technician got fed up and refused to feed them. But no one yet knows how stable they are in living beings, nor how long they last. Scientists do know, however, that senescent cells are found in people of every age from babyhood upwards, and that they are normally recognized and cleared away regularly by the immune system. But as we get older, so too our immune systems age and get less efficient at clearing senescent cells, just as we are producing more of them. Thus, senescent cells accumulated in our tissues as the years advance, clustering particularly at sites of so-called age-related disease such as skin, ulcers, arthritic joints, and compromised arteries. But does this completely explain why cells die? Today will be just another day unless you decide today it won't be. It is Tuesday, July 30th, 2019. Let's flow.
So, my name is Judy Campisi, and I thought Aubrey was going to say we're going to start at the lower end of the spectrum, and that's why I'm first. Thank you. Thank you. Okay, so I have a big question mark here, and I'll come to that at the end. But so I assume everyone here wants to live a long life, right? Any exceptions? Good. This is what you have to look forward to. You know that, right? These are the diseases of aging. What is so remarkable about these diseases of aging, typical diseases of aging, very different tissues, very different cell types, very different processes that cause the pathology. And yet what all of these have in common is this: very rare in young people, and somewhere around the midpoint of the lifespan, all of these diseases rise with approximately exponential kinetics. Now, I would say that there's consensus now in the field of aging research that this is not a coincidence. Of course, it's possible. It's formally possible that each tissue has its own trajectory of how it increases in pathology with age. But most of us believe that this is not a coincidence and that there are a few, we don't even know how many, but a few basic aging processes that are driving multiple age-related diseases. And the beauty of that concept, if we're correct, is, as Aubrey kind of alluded to, it would really revolutionize the way medicine is practiced, because now there would be physicians who would be treating aging and not treating cardiovascular disease or cancer or macular degeneration, independent of each other. So I'm going to tell you a story of a possible basic aging process that we think we've discovered and have accumulated some evidence that, at least in mice, this is the case. So this is the hypothesis, and the process that we think may qualify for being one of these basic aging processes is a cellular stress response that has been termed cellular senescence. This process was first discovered in the 1960s. It was really not understood very well, but it's been studied since that time, and we now know a fair amount about the cellular and molecular sequela of cells undergoing this stress response. And I'm a little bit oversimplifying what the stress response is by telling you that we think it's basically a bipartite phenotype. The first part of the phenotype is cells stop dividing. So this is unique to cells that can divide. This is not something a neuron goes through, but this is something that epithelial cells or stromal cells or endothelial cells go through. And what they do is they lose the ability to undergo cell proliferation. So almost from the very beginning, actually in the 1960s, it wasn't even appreciated so much, but it has been now over the last several decades, that this growth arrest is critical. It is the reason, or one of the important reasons, why cancer is one of those age-related diseases, very rare in young people, rises exponentially at about the midpoint of the lifespan. So we have mouse models, and there are people with mutations that prevent this growth arrest, and they die an early death due to cancer. I'm going to come back to this point that this is good, the fact that cells undergo this irreversible growth arrest. But coupled to this arrest is something that we've termed a secretory phenotype. And what that means is that the cell that has stopped dividing now begins to make molecules, bioactive molecules, that influence the neighboring cells. And I'll show you some examples of how that occurs. So this bipartite phenotype, now we know, can be induced by many, many types of stresses. So some is the kind of damage that Aubrey was talking about. Genomic damage, epigenomic damage, the activation of many oncogenes will cause, that makes sense, right? If it's an anti-cancer mechanism, you activate an oncogene, the cell stops dividing. But we now know metabolic imbalances, organelle stress, for example, cells that are stressed by secreting too much, all of these stresses can elicit this bipartite phenotype. And the way we think of it, though, is that it is an evolutionary trade-off. That is, that the good part is to prevent cancer, and I'll mention to you some recent data that suggests that these secretions can also help tissue repair and remodeling, especially wounding. Balanced against that are these phenotypes that we recognize as aging, and virtually all of those age-related diseases that I showed you on my first slide. So this is going to be tricky, because you want to maintain the good while ablating the deleterious effects. We think we can do that pretty well in a mouse. We have no idea whether we can do it in people. So over the years, many labs have studied this process of senescence, and we now have a handful of markers, biomarkers. I do want to emphasize we have no specific senescence markers, so we tend to look at the whole phenotype and then hope that we have some confidence when we see cells that we can identify them as senescent. So using these markers, I want to emphasize two of these markers. One is this protein called P16. This is a tumor suppressor protein. It stops the cell cell cycle. It's part of the reasons why the cells stop dividing. And I'll show you that we can use this protein to generate tools to study this process in mice. The other point I want to make is the cells become resistant to death. And we think this may be part of the reason why they accumulate with age. So that gets to my second point, which is using these markers now, we can ask when and where do senescent cells appear. First is they accumulate with age. This has been shown in virtually every vertebrate species that has been studied, and now there's at least some credible reports in Drosophila that at least in the gut where there is cell proliferation, there's an accumulation, age-related accumulation of senescent cells, and it's been shown in virtually every tissue that has dividing cells as part of its compartment. The second place we see them is in pathology, age-related pathology. Both the degenerative pathologies, that is the pathologies that are marked by tissues failing to function as well as the hyperplastic pathologies, the pathologies that give rise to cancer. So cancer is very different from the degenerative diseases. So here are two examples. This is COPD, chronic obstructive pulmonary disorder, major cause of lung failure in the elderly. If you compare the number of senescent cells in the lung of diseased patients versus non-diseased patients, there's a significant increase in disease. These are age-matched. And the same thing is true if you look in the brain. So we tend to think of the brain as being post-mitotic. It's not true, actually. The major cell type in the brain is the astrocyte, more astrocytes than neurons. Astrocytes, of course, give rise to brain cancer. And what you see in diseased brain, in this case Alzheimer's brain, is more senescent astrocytes compared to age-matched controls. So I'm going to make this is a very American slide, right? You can say that senescent cells are a smoking gun, right? They're present at the right time and the right place to be driving these many diseases that we think are glued together by a single process. Now, the two questions, of course, that we grappled with when we began studying this is, first of all, how? How could these cells be driving such diverse pathology? And the second is, do they really do it? Because everything I've told you so far is correlation. So let me start with the second question and tell you why or how we think senescent cells might be driving pathology. And we think the key is this stuff that they're secreting. So these secreted molecules include cytokines, chemokines, growth factors, proteases, bioactive lipids like prostaglandins. And what they all have in common is they have the ability to drive inflammation. And inflammation is the commonality behind all of those diseases I showed you on my first slide. Inflammation either causes them or greatly exacerbates them. And we know what inflammation can do. It can destroy tissues, literally cause them to fall apart. It can disrupt normal tissue function. It can prevent stem cells from functioning properly. And of course, it's a big risk factor for the development of cancer. So I'm going to show you 3 examples of how what senescent cells secrete can have effects not on themselves, but on their neighbors. And the first example is in the mammary gland. You can take cells from a mammary gland, put them in culture, and if you give them the right hormones, they make milk. So the red staining here is these epithelial cells that have organized into alveoli and are now producing milk. And if we embed in these cultures non-senescent cells, we get these beautiful alveoli. And senescent cells, really, you get ugly alveoli, they're misshapen, and also they're not functioning because they're not producing as much milk. We know the factor that senescent cells produce that causes this destruction, and it's mostly the proteases that the cells produce. The second example I'll show you are stem cells. So what you're looking at are human neural stem cells, and the green is their ability to synthesize DNA. So that's one of the most important things a stem cell needs to do, is it needs to divide to replace itself and then replace the tissue. And senescent cells make molecules that suppress the ability of stem cells to divide. The third example I'll show you is something that was really shocking when we first saw it. So what you're looking at is tumor volume over time, and what you can see is that we've injected into the mammary gland of a mouse pre-malignant epithelial cells. So these are not cancer-forming cells, but they're not normal. They have some mutations, not enough to form a tumor. And if we co-inject them with senescent cells, now we get full-blown conversion to malignancy. So the model then is that with time, we accumulate senescent cells, and this is critical for keeping us cancer-free for about half of our lifespan. But these senescent cells now will affect their neighbors and cause degenerative pathology. And of course, what really happens with age is we're all accumulating premalignant cells, and we're now arguing that senescent cells can even drive those premalignant cells to cancer. So if you're not depressed, you haven't been listening.
Now, here are my 2 cents, and feel free to keep the change. I'm not a believer in so-called age-related diseases, but are there diseases that are more likely to manifest as we age? Of course there are. Our cells take many hits over the course of our lives, biological hits, if you will, such as colds, diseases, viruses, etc. And those hits over time have a cumulative effect on how the cell communicates with neighboring cells, as well as how it behaves. But my question is, are these a result of aging, or are they due to the choices we make throughout the process of age? One of the major takeaways is the role inflammation plays in nearly every degenerative and so-called age-related disease. So is the buildup and proliferation of inflammation a more accurate assessment of why cells die? As noted, inflammation certainly plays a major role, but still it isn't the only determining factor. I'm always amazed at how diverse and resilient our bodies are, indeed how amazingly durable our cells are relative to the deceptions we put them through. And what do I mean by deceptions? Well, think about how many times you subjected your body to alcohol or drugs or malnutrition or voluntary starvation in the form of radical diets or dehydration or sleep deprivation or fast food binges or sugary drink substitutes, voluntary cosmetics, just to name a few. And yet you convinced yourself it wasn't a big deal because you didn't put on a lot of weight, or that the cosmetic surgery was non-invasive, or that you only drank casually, or that popping OTC pain relievers were not really drugs, or that coffee and other beverages contained water, or that you were only dieting to get into that bathing suit. Any of these deceptions sound familiar? I'm sure they do, but here's the thing. Your body doesn't distinguish between rationalization and reality. What you feed it ultimately determines what it believes you want. In other words, your body is highly adaptable to everything you subject it to. Thank goodness for that. Its adaptability is perhaps the only reason you're still alive, to abuse it. Dear friends, I wish above all things that you prosper just as your soul prospers. You've been listening to Urban X Real Talk Fitness Radio with your host Tiaja. Until next week, as always, walk in health and peace.